A very good morning to one and all. Thank you for joining us on the third day of this AICT sponsored faculty development program under AICT Training and Learning Academy on emerging trends in drug engineering. On behalf of entire principal KM Kundanani College of Pharmacy, I, Ms. Ruchira Sarunkhe, Assistant Professor in Pharmacy, feel privileged to welcome all the delegates present over here. Further, I would like to extend my warm welcome to our principal, Dr. Urmila Joshi, ATAL FDP coordinator, Dr. Mrinal Sanai, all the chairpersons and the faculty of KMK College. To begin with, I would like to request ATAL FDP coordinator, Dr. Mrinal Sanai, to brief us about yesterday's session and give us preface for today's session. Please, ma'am.
Yeah, thank you, Ruchira madam. Thank you for that wonderful welcome. Dear participants, again welcome back to our day 3 of ATAL FDP Emerging Trends in Drug Engineering. And today we have an opening session with Dr. Subhedar, who is a visiting faculty in IISER Pune. But before we start with the session, I will just take a review of what we have done yesterday. There was a very beautiful session by Dr. Samir Kulkarni, who has in a lucid manner explained us a very critical process of development of biosimilars. And then we had a very interesting session by Dr. Rajesh Kumar Goel, who actually demonstrated, also shared lot of information on the network pharmacology. After that, it was a very nice session covered by our own alumnus and a very well-known professor, that is Professor Vandana Patravade from Institute of Chemical Technology on co-crystallization. Then there was a session by our principal madam, Dr. Urmila Joshi, on kinase inhibitor, where she explained all the details about the kinase inhibitors in a very interesting manner. And today we are also having a very interesting sessions, as I already told you, that we are opening with a session of Dr. N.K. Subhedar sir, who is known as a Bhishmacharya in the neuropharmacology. And it was a long awaited dream of KMK to have him, to hear from him and get the knowledge on neuropharmacology from him. And then we are having a session by Dr. Deepak Thakur, who will be speaking on mass spectroscopy characterization of various biotherapeutics. After that, there will be an interesting session by Dr. Surajit Debnath, where participants will get an opportunity to directly interact. It will be a very interactive session with some workshop, which details of this workshop are already mailed to you, and I hope all of you have downloaded all those links. So let's begin with our next day of the journey of knowledge with Dr. N.K. Subhedar sir. Sir, welcome to our FDP.
Thank you very much, Mrinal Sanai madam, for those kind words. And I'm really overwhelmed for two reasons. I have heard of this your college for a very long time, and I always wanted to visit, and I'm happy that today I get the opportunity to meet you and your faculty and your participants, and I think this is great. And secondly, I find that you have had a series of star speakers, and the topics they covered are really very relevant today. And I'm absolutely sure that the participants have extracted every word of wisdom that the talks could give. And it's really very nice. And I would again like to thank all of you and address the principal Urmila madam and Mrinal madam and the rest of the staff. And if with your permission, I'll let us begin our scientific endeavor. And have really been looking forward to presenting this data in front of the committee before this audience. So can we just go to the title slide of the talk, please?
Yeah, madam. Sir, I will first introduce you and the speaker, and then we can begin with the.
Please. Sure.
Okay. May I now like to request Dr. Mrinal Sanai, Associate Professor of Pharmacology, in charge Head of Department of Pharmacology, to introduce our first speaker for today, Dr. N.K. Subhedar sir, who will deliver the session on advances in pharmacological screening techniques. Please, madam.
Thank you, Ruchira Madam. It's an honor to introduce Dr. N.K. Subhedar Sir, who is currently associated with IISER Pune and teaching animal physiology 1, 2, and neurobiology. He has worked from 1972 to 2008 as a faculty at the Department of Pharmaceutical Sciences, Nagpur University. And he retired as a professor of pharmacology in year 2008. There onwards, till year 2018, he has served as a visiting faculty at IISER Pune. 2019, he was also teaching animal physiology at Ashoka University, Sonepat. 2015 to 2020, he has taught a course of neuroanatomy at JNCASR Bangalore. He has lot of grants at his credit. Some of them to mention, he has received a funding from DST, DBT, ICAR, UGC, IBRO, TWS. He has an enriched experience of guiding research students, and I have personally seen that the way he gets bonded with the students during his research work is extremely appreciable. He is a PhD guide at Nagpur University and he has guided around 15 PhD students. His areas of research include the role of neuropeptide transmitters in processing hunger, anxiety, depression, learning and memory, reward and drug addiction related information. He is also working on study of alpha melanocyte stimulating hormone via MS4 receptors in ethanol induced anxiolysis and withdrawal anxiety, probing the role of peptide, cocaine, and amphetamine regulated transcript in early signs of anxiety-like behavior induced by ethanol withdrawal within the neuroanatomical framework. He is working on study of neuropeptides acting Y1, Y5 receptors and their influence on processing of a neuropathic information. He is studying on CART peptides in the processing of fear and reward in disparate brain circuits. He has around 130 more than that research papers to his credits. He has written one chapter in a book and two reviews to his credits, and general articles and book reviews to his credit are 5. He is an ad hoc reviewer of several international journals. He has delivered many talks in several institutions, universities, India, as well as abroad. He has participated in several scientific meetings in India and abroad. He is a reviewer of a research projects for SERB, CSIR, UGC, DBT, Portuguese Foundation for Science and Technology, Erwin Schrödinger Fellowship, Austria. Sir, welcome to our session. It's really an honor, and we will start with a scientific session. I'll just share your presentation. Is it visible, sir?
Yeah. No, we have to go to the first slide. Yeah. Great. Yeah. Full
So,
screen mode.
yeah.
Or
You
this
just
is fine for me.
go to next slide. I will do that.
Good. So good morning once again, and thanks to Munara Madam for that a little overwhelming introduction, but that's okay. Okay, so before I begin, let me briefly tell you as to what we have been doing all these years. We have been interested. First of all, we are interested in brain, and we use rat and mouse as the model. And our interest is mainly focused on the neuronal circuits. In brain we have different groups of neurons, and they are talking to one another. And as the information flows over them, the different functions emerge, and they help the animal to survive in the environment. And if something goes wrong in that circuits, then the different pathological conditions emerge. We have been interested in some of the neuropeptides which serve as neuromodulators and neurotransmitters in the brain, and we have been trying to find out as to what are their functions in the different areas. Now, before I there's one very interesting peptide in the brain which is called as CART, abbreviated as CART. The whole name is cocaine and amphetamine regulated transcript peptide. A biggish name, complicating, but it's very simple actually. I'll tell you the story. What happened in about 1990s or so? When they injected the rat with the amphetamine or cocaine like these drugs, they found a new peptide. And because that new peptide was discovered in the animal that was injected with cocaine, therefore they gave this name, cocaine and amphetamine regulated transcript neuropeptide. And now there has been a lot of papers in this, and and one of the reasons why there is considerable interest in this peptide is because this peptide is very widely distributed in the brain of all animals, including you and me. So in our brain, there is plenty of neuropeptide. It's there in the cerebral hemisphere. It is there in the cerebellum. It is there in the thalamus, in the hypothalamus, and we are just beginning to realize as to what are the different functions the peptide may be undertaking in different parts of the brain. So if I go to the next slide, I'll briefly tell you some of the functions which are very well established. Very good. Now to show you the chemistry of the peptide, the propeptide is actually very long molecule, about 1 to 102, but eventually the molecule is cleaved and the active form is called as CART 55 to 102. So this fragment which is shown here, I have copied this from a research paper by Rogue et al. And this is more or less the structure of the neuropeptide. And we know very well that this peptide is abundantly present in the hypothalamus. Now what is this in front of you is a slide of a section through the hypothalamus. And in the hypothalamus, you'll find here, there are different neurons, and I'll draw your attention to this neuron, which contains two peptides. One is called as POMC, pro-opiomelanocortin. It's a peptide, and it also contains another peptide, our familiar CART. Now, what is the peculiarity of this peptide? This cell, I'll tell you. It's very interesting. There are two sets of neurons in the hypothalamus. Well, there are many, but let us talk about two. Some of them are containing this peptide, pro-opiomelanocortin, POMC and CART, and others are containing AGRP and neuropeptide Y. So there are two sets of neurons. Some are containing this neuropeptides and some are containing this neuropeptides. Now just imagine you are starving. Okay, you have not had food in last 3 hours or so, and you are hoping to have some lunch. At that time, one interesting thing happens is that your stomach starts releasing a protein hormone, and that protein hormone, which is called as ghrelin, that starts acting on these neurons, and under the influence of ghrelin, these neurons secrete neuropeptide Y and AGRP. They go to different area of the brain and the hypothalamus, and they generate hunger. Not only they generate hunger, they trigger your behavior so that you will start looking for food, you will try to consume food. Now, let us imagine a situation that now you had the food, you had access to food, you had food, but then you don't keep on eating. You see, after some time, when your belly is full, you tend to stop eating. That happens because from the the insulin from your beta cells start secreting insulin. Now insulin is suddenly in high concentration. It is triggered by the glucose in your blood, and that insulin and leptin, these two hormones of the periphery, leptin is coming from the adipocytes. These two hormones have an effect on these neurons. So CART neurons, when they get information from insulin and leptin, they get excited and they carry the message to yet another group of cells, and they give a message to you, please stop eating. Therefore, we say that no, I have had enough, and we give the information to the brain which says satiety. Now we are satisfied and we don't want to eat anything more. So we know that CART has a very important role in inducing satiety or telling you when to stop in weight. So therefore, it regulates your behavior in asking you to stop here. So it's a very critical peptide. So I'll introduce to these two peptides. They generate hunger and they drive you to food. But when you have enough food, these two peptides eating, and so that you don't overload your system. Now, so what I did just now is to tell you one very interesting function of the performed by the CART, and this is very well studied. You will find hundreds of papers on this line. But this peptide is also occurring in other parts of the hypothalamus, of the thalamus, of the olfactory system, and in the amygdala. And what is the function of this peptide in those areas is still a matter of a bit of mystery, and serious investigations are in progress to find out what it is. Now I'm going to do change of gears. Now I want you to hold to the thought on the CART peptide. Just hold on to that thought, and now we'll switch the gears and I will show you something equally interesting. Next, please, ma'am. Oh, this is a very interesting picture I want to draw your attention to. I am taking you back into the history of science, and the year is about 1954-55. We are somewhere in McGill University in Canada, and this is a group of scientists here. And I'll tell you, his name is James Olds. Here is another scientist by the name Peter Milner, and these two are actually psychology students. Psychology. One is a faculty. James Holt is faculty, and Peter is a postdoctoral student, and they are working on the rat model. And they are psychologists, so they want to find out if they are asking. Their question is, if I place an electrode in a certain area of the brain of rat and give an electrical shock, does that make my animal smart? Can I make my animal more intelligent? And how do I test whether it is intelligent or not? Then I let it run in a maze. So in a maze, it's a complicated pathways that we all know what a maze is, and they hide the food in new places, and they want to find out whether my rat, after electrical stimulation, is more smarter in finding the food. So when they are doing an experiment on this animal, where you can see in the picture, it is just an arena, and they realize that this particular guy, this particular rat, when the animal in a particular corner of they gave a shock, they found that now they are not giving the shock, but the animal was repeatedly returning to the same corner. A phenomena which we call as place preference, something like condition, something like that was happening, and the animal was repeatedly coming to the same corner. So they started asking a question: is there happening something in the animals so that the animal liked the shock? And therefore it was, and the place where it got the shock, it wants to repeatedly visit the place so that it can again experience that happiness. That was a question that came to their mind. Now, having got that thought in the mind, they were very excited about it, and then they tried to they repeated the experiment on this very animal, and then something very interesting happened. When they killed the animal and they opened the brain, and they found that their electrode was not in the hindbrain, but it was actually located in the hypothalamus. So they were targeting the electrode somewhere, the electrode went somewhere else, but by this accident, they now realize that there is a place in the hypothalamus where if you electrically stimulate that place in the hypothalamus, then the animal seems to experience something, happiness, some pleasure, and therefore it is returning to the same place. Now, with this background, they did something more interesting. Next slide, please, ma'am. Okay, now considerable time has passed. Then in 1960, 70, more research was done, and then they realized that the further experiment showed that there is a certain relationship. What are the point where you are stimulating? The point where they were stimulating is an area which is called as ventral tegmental area. What are we talking about? We are talking about a group of neurons. Where is this group of neurons located? In the midbrain, in the lower part of the midbrain, therefore the tegmental area. And what did they find? They found that if you electrically stimulate this group of neurons, then it excites another group of neurons here in the group of neurons called as nucleus accumbens. Now, I will request you to remember the two names. They are very critical to my paper and to your general understanding of the brain circuit. One is ventral tegmental area. So they are electrically stimulating the ventral tegmental area, and in the nucleus accumbens they have inserted a probe, and through that probe they are drawing the fluid there, and they are analyzing the fluid for the content of dopamine, and they find that, now I'll draw your attention to this graph here. This pinkish band indicates the time during which they are stimulating the ventral tegmental area, and in this time the blue curve indicates the amount of dopamine. So when you stimulate in this part, you get a peak in dopamine, then don't stimulate for some time, then again you stimulate, again you get a release in dopamine. So the circuit was discovered that there are neurons in the ventral tegmental area. If you excite them, they are responsible for releasing certain dopamine in the nucleus accumbens. Now, how is this relevant? Next, please, ma'am. Okay, now we know, we have studied more. I will take you to about 1990. we now know, now this is an image from Poxinas and Watson, which is the stereotaxic atlas for the brain of rat. And here you get a very small, it's actually not a big neuron, it's very small, it's very easy to miss if you're not careful. But if you stain it for dopamine containing cells, you distinctly get a group of neurons, and that group of neurons is diagrammatically shown here. We find it in the posterior ventral tegmental area, and we now know, the studies have shown that it has connection to the accumbens nucleus that I have shown in the previous slide, and it also has extensions to the prefrontal cortex. And this now is identified as the basic anatomy of the circuit, which, if excited, is known to generate certain happiness or certain reward. And if that happens, then animal works more and more to generate that experience. And how that happens, let us please see in the next slide. Okay, now what you do here is you place an electrode in the ventral tegmental area, and then you place that electrode, that electrode is connected by this wire, and this wire then goes to the outside, and the wire is connected to the liver. So you can see here there is a liver. Okay, now and we'll call this as an operant chamber, or a chamber wherein the animal by trial and error, it learns. What does it learn? It learns, or you can teach the animal that if you press the lever, you are actually starting a circuit. That circuit will give a shock in certain areas of the brain, in the hypothalamus and in the midbrain, and that shock is very fond. The current is very in very weak strength, and if you do that, then the animal, if it feels good, then the animal can be trained to press the lever repeatedly, and then the animals, and if the trained animal likes it so much that the animal will keep on pressing it. And even if you give the food to the animal will prefer to press the lever and not go for the food. So as if the animal has realized that pressing the lever is a direct shortcut to my happiness, and if that shortcut is there, I don't need to go to other sources like food. Now here in this particular case, you can see those red lines at the bottom. So what did the experiments do? That they lift the animal on this side, on the other side, and the liver is on the other side. So those red lines indicate that there is electrical shock. So the animal, now this is a trained animal, the animal wants to go to the liver, but if it has to go, it has to suffer the shock. The animal doesn't care. The animal just runs over the grid which gives him electrical shock. It suffers the shock, but it goes to the other side and it keeps on pressing the lever so as to experience the certain amount of reward that is directly being delivered to its brain by way of the electrical shock in specific areas of the brain. Next, please, ma'am. Now let us see what is it that you see, the nature has given this pathway. This pathway is in the rat, it is in the mouse, it is in your brain, it is in my brain. And whenever so when you are hungry, you are looking for, you say you are thirsty, you are looking for water, and then you get a glass of cold water and you like it. At that moment, and you like it, and because you like it, you take one more sip. That is happening because the ventral tegmental neurons in the ventral tegmental area of your brain are excited. They take the message to the nucleus accumbens. They release dopamine in the nucleus accumbens, and therefore you have the pleasure of drinking water. So you associate that pleasure with a glass of drinking water. Same thing happens when you have food. You are hungry. So let us see, here is an animal which is hungry, which is there's no food. It has been starved for some time, and then you suddenly give food to the animal, and what you are doing is you have put a probe in the nucleus accumbens. From there you draw the sample and you analyze the sample for amount of dopamine, and you find the dopamine content has gone up, and after some time it will go down. But this peak in the release of dopamine is because the animal was suddenly given food. To the starved animals, the animal was suddenly very happy, and that was displayed by the and the underlying principle for that is the release of dopamine in the nucleus accumbens in the prefrontal cortex. Now this is, you see, whatever is the source for pleasure, whether it is you are thirsty, you drink water, you are hungry, you have food, or your sex, or your friend cracks a joke, whatever is you suddenly you like a piece of music. So whatever experience that you come across, and if you happen to enjoy it, mind you, the basic neural circuit that underlies that experience, it starts with the ventral tegmental area. It takes you by way of dopamine to the nucleus accumbens. So here's a very interesting experiment that was undertaken on the male rat. What they did was, there was a male rat that was sex starved for some time, and there was a curtain, and on the other side of the curtain, a female rat was placed. The male rat could not see the female rat, but of course, for the rat and for the rodents, the sense of smell is very important. So the male rat suddenly got the pheromones from the female across the curtain, and when it got the pheromones, and what you are really doing is you are drawing the sample of fluid from the nucleus accumbens of the male rat, and you find that the dopamine concentration has doubled from this 100 to 200 units. So there was sudden burst of dopamine the moment male rat could get these smell of the pheromone that was released by the female. So whatever is the underlying reason, whether it is water, whether it is food, whether it is sex, or whether it is whatever it is invariably the excitation of this pathway, which starts from the ventral tegment area and nucleus accumbens, is critical to generating generation of pleasure in all mammals. Can I have the next slide, please? Now, this is our knowledge of the pleasure pathway. It starts with James Olds in 1953-54. We can celebrate 70 years of our information or of our knowledge and wisdom on the pleasure pathway. But the human beings realized about 300, 3,000, 4,000, 5,000 years back that you can excite the pathway. Of course, they didn't know what the pathway is, but you can excite the pathway by using drugs. And soon humans knew about nicotine, they knew about opium, they knew about alcohol, and they have known it for centuries and centuries together. So what is really happening is, it was of course by chance that humans, those days, they realized that these drugs can directly excite those particular pathway. When I say those pathway, I mean the same pathway which starts in the ventral tegmental area and goes to the nucleus accumbens. And if that pathway, so when you are actually taking nicotine, you are exciting that very pathway. So we realized it, but this was also a point for alarm bell. Because these drugs, which are of course not natural, food is natural, sex is natural, water is natural, but these drugs are not natural. Over excessive uses of these drugs, they interfere with this pathway which I talked about, and they remodulate the pathway, they change the pathway, they induce plasticity on the pathway, and as a result of that, when you want to discontinue with the drugs, it's a little difficult because now you are addicted. So here is a classic example where not only humans, even the chimpanzees, this particular chimpanzee that we are seeing has been hooked to smoking and has been hooked to nicotine. And now we know, recent researches have shown that the pleasure that you get after receiving money also gives rise to the excitation of the ventral tegmental area, nucleus accumbens pathway, and the release of dopamine. Next, please, ma'am. So we have, I have drawn in front a very simple slide from the brain of frog, and here is the ventral tegmental area, here is nucleus accumbens, and let us see the different, there are receptors wherein opiates can act, ethanol can act, barbiturates, benzodiazepines, and nicotine. So all these drugs, which we know very well, they are the drugs of abuse. They are acting at this particular site, and other drugs like amphetamine, cocaine, opiates, cannabinoids, and phencyclidine. These are drugs are acting directly on the nucleus accumbens. Okay, and the excitation of the neurons here by these group of drugs, or excitation of neurons in the ventral tegmental area by these bunch of drugs, they are capable of directly exciting the pathway. They generate pleasure in the brain, and eventually they modify the circuit, and once that circuit is modified, you get fully addicted. And then of course, then as pharmacology students, we know that if you withdraw the drugs, we have withdrawal symptoms, and you know all the complications, and you also know why withdrawal is so easy and relapse is by giving up the drug is so difficult and relapse is so easy. Okay, please, ma'am, can I go to the next slide? So here we have a list of we know what are the agents that can, of course, nicotine is there, amphetamine is there, cocaine is there, heroin, alcohol, opium is there, even food. Even food can generate, there are other range of banned drugs. Look at this very interesting image, even gambling, okay, dealing with cards. They all are addictive and they can give rise to, they reshape your circuit. And these days, I'll draw your attention to the problem with our younger generation, which is facing is, if you see here a young boy and girl having good time with one another, no, you're wrong. Each is busy on its own cell phone, and we all know how addictive the cell phone is. Please remember, cell phone is addictive because excessive use of cell phone is also introducing plastic changes in the circuit in your brain, which is going from the ventral tegmental area to the nucleus accumbens and the prefrontal cortex. So excessive use of the cell phone, you're also subjecting yourself to the remodeling of your circuits in the brain. And once that happens, well, you know what happens. Next, please. Okay, now I'll, for a very long time, there have been studies on finding out which are the different areas where you can place an electrode, and once you play in the rat brain, and once you place an electrode, if the animal is able to if the animal can be taught to self-stimulate, then you know that the area is in the pleasure area. Or let me put it other way around. If you place your electrode here or here or here or in this area, please remember, if you place your electrode in specific areas, then you can take the animal to the operant chamber. You can connect the animal's electrode, which is in the skull of the animal, to the liver out to the circuit outside, so that when the animal presses the liver, the circuit is on. And if the animal likes it, please remember, if the animal likes it, then only the animal can be trained to press the liver repeatedly. By that I mean, if you place your electrode in the cerebellum, in the cortex, here, anywhere, if you place the electrode, then the electrode is in wrong area. The animal cannot be conditioned. You cannot train the animal. No matter how much shock you give, whatever, the animal will never learn, because getting shock in that area is not generating any pleasure, and then it is pretty useless, which means you have missed the target. So one of the very, very basic features of this experiment is that you must place your electrode either in the prevent. There are a series of areas which have been defined, and we are now concentrating on this particular area, which is called as LHMFB, lateral hypothalamus medial forebrain bundle, which is one of the most preferred areas where people place an electrode, and then the animal can be readily taught. And this is exactly what we have done in our lab. In our lab, we place an electrode in the LHMFB area, and so lateral. Let me define again. Lateral hypothalamus medial forebrain bundle. What is this? This is an area. Where is it? In the lateral hypothalamus. Where do we have? We have pathways, and we have neurons. And if we place an electrode there, then the animal can be readily conditioned to self-stimulate, and the technique is called as intracranial stimulation. This is fairly well recorded in the literature. So what are we doing in this slide? If you are able to stimulate the neurons in the lateral hypothalamus medial forebrain area, then the information goes to the ventral tegmental area. In the ventral tegmental area, there are this red refers to the dopaminergic neurons. When they are excited, the dopaminergic neurons release dopamine in the nucleus accumbens. When that happens, there is increase in the release of dopamine, and that gives rise to generation of reward. Now, this circuit is again very well established in literature. What is not known is what kind of neurons are they. Are they GABAergic neurons? Are they glutamatergic neurons? Are they NPO? What kind of neurons are really involved in carrying the information from the lateral hypothalamus to the ventral tegmental area is not really known, and therefore we have been interested in focusing our attention on this kind of studies, because we are interested in finding out more and more about the reward circuits in the brain of the rat model. Next slide, please, madam. So what we have done in our lab is we have placed an electrode. So okay, so what do? We anesthetize the animal, and then we remove the skin, and then we are using stereotaxic atlas provided by Paxinos and Watson. We determine the precise spot on the skull where we drill a hole in the skull, and through that hole we introduce what you call as an electrode to a certain depth, which is again guided by the atlas by the Paxinos Watson, and you can find that your electrode has been introduced so far. And then eventually, when the experiment is done, we have taken the sections through the brain of the animal. So this is one section through the brain which goes through the hypothalamus. So this is the section through the hypothalamus, and I'll draw you. Can you see this white line here? Okay, this white line is because that was the passage taken by the electrode, and the electrode was introduced right at this spot. So this is just a confirmation that your electrode has gone all the way to the medial forebrain bundle, and that your electrode is in small place. Then this is an animal where you can see the electrode, and the electrode is now secured on the skull with the help of the glue, etc. And you have a bipolar electrode, and that bipolar electrode is now connected by way of this wire. This art and science of making an electrode has been standardized in our laboratory. It's already published. For those who are interested, I can share that information as to how you can make electrodes in your own lab. You can import them from outside, but they are very expensive. So it makes sense for us to make our own electrodes, and that literature is published somewhere. And if somebody is specifically interested, I will let you know those references. So here are the electrodes which are implanted and secured on the skull of the rat, and the rat is fully conscious. It has come out. So you place an electrode, you give 7 days to the animal. So the animal is now recovered. It is doing all its activities normally, and then when you want to do the experiment, you connect the wire to the electrodes. That wire goes to the outside. It comes out from the roof of the operant chamber, and then it is connected to the apparatus, and that apparatus is actually it's a stimulator, and that stimulator is now connected to a lever, and that lever goes inside the operant chamber. Now inside the operant chamber, we actually provide 2 levers. One lever is such that anytime animal presses the lever, the lever goes back, and then it comes up. So every time it goes back, it is recorded as to how many times. So animal presses, the lever goes back. You also provide a very similar another lever, and that another lever we call as inactive lever. Means what? If animal presses the lever, nothing happens. Okay, so if I'm doing an experiment and if it is not generating any pleasure, then you will find that the number of presses of active and inactive lever is the same. It means there is no difference between the animal is not getting any pleasure or any reward. So we always give an active lever, which if pressed by the animal will get a shock. And if the animal likes it, the animal will repeatedly press the lever, which means the animal has been now conditioned or is now trained to repeatedly press the lever so that it can generate happiness within the brain. So what we have really done is we have altered the behavior of the animal into trying to get access to the lever and press it repeatedly. And if the animal does it, because every time it does, it gets a shock, and that generates a certain amount of happiness. So this is just a photograph of in our lab showing the rat in the operant chamber. And next, please, ma'am. Yes. Now, before you go to the video, ma'am, what we have done is we have taken a small 31-second video in which you will see that the rat is in the operant chamber, and how the rat is excessively engaged with the lever. Only in the active lever, because it is pressing of the active lever. So what we now something when you can also consider it as a serendipitous observation. We found, you see, we have been working on the cart containing neurons in the different parts of the brain, and particularly on the hypothalamus. we found that this lateral hypothalamic area, where if you place an electrode, the animal can be conditioned, we suddenly realize something interesting, that this area contains a lot of CART neurons. So this is a section through lateral hypothalamus which is very rich in CART neurons, and this is also the area where if you place an electrode, you can see the site of the placement of electrode. So this is actually the area which is containing a lot of CART neurons. The CART neurons have been stained with immunocytochemistry using antibodies against CART, and we found an abundance of CART neurons. So we started asking a question: is that electrode? So this is a question, we don't know the answer. We ask a question: is our electrode, which is generating the behavior of self-excitation, is it via the activation of the CART neurons? Yes or no? We were trying to find out an answer, and therefore we now conducted a series of experiments to test if in the animal that has been trained to self-stimulate, what is the response of the CART cell? Do they have any involvement in the rewarding behavior, was a question we are asking, and therefore we conducted a series of experiments, and the results of some of the experiments are in the following slides, please. So we test the hypothesis. CART containing neurons in the LHMAB region have projections to the VTA and positively modulate the mesolimbic dopaminergic reward pathway. It is the same pathway that goes to the nucleus accumbens. So we want to test this hypothesis, and therefore we plan the following experiments. Next, please. Okay, so these experiments were actually done by Amita in our lab, and what Amita did was, you take these animals, you train them extensively. Every day for 30 minutes you let them press the lever. Okay, you let them do it for 1 or 2 weeks, and after that you simply okay, now please remember, the electrode is on one side. So on one side, you don't have to put 2 electrodes. It's unilaterally you place an electrode on one side which goes into the LHMAB area, and you condition the animal. Now animal is very well trained. You leave the animal there, and it will go to the liver and it will start pressing the liver. What you do, you call that side as the ipsilateral side, and you call the other side as the contralateral side. So when you have the animal which on the ipsilateral side, means this is the LHMAB area, so you take sections from this part of the brain and you stain for the CART neurons here, and you find that there are so many CART neurons there. As against that, in the contralateral side, there are very few neurons. And this is the LH area of the control animal, which is not conditioned. So in the unconditioned animals, there are so many neurons, but in the excited animals, there are so many neurons. Means the number of CART neurons in the animal which has been trained, where a number has gone up, and you can see the difference. The readings are actually in front of you. Second thing, what she did was, she also co-stained the cells with another antibody, which is called as antibody against CFOS. Now, many of you would know that CFOS represents a protein that comes from immediate early gene. It is an oncogene, and this is a very useful tool to find out whether the neurons are activated. So, and this is a nuclear stain, so you can see every red dot shows a nucleus, and here you can see that some of them are co-localized. Means here we can say that not only more and more neurons are expressing CART, but more and more neurons are expressing CFOS. Means, and I want you to compare with this, less CFOS, less CART cells in unconditioned animal, more CFOS, more CART on the ipsilateral side, but less CART and less FOS on the contralateral side. So on the side where electrode is placed, you find that there is more CART, there is more CART synthesis, there is activation of the CART cells, and therefore we can say that it may be directly responsible to the electrical current that you have delivered directly into the lateral LHMAB area, and the CART neurons are specifically responding to that. Can I have the next slide, please, ma'am? Having done that, they see the CART protein is going up. So we thought, is the CART should normally be synthesized. Okay, so to do that, what we did was, we wanted to look at CART mRNA. So this experiment was done by Neha. What she did was, she did something similar to what Amita did, but this time she took the entire brain. From the entire brain she collected this tiny fragment of brain tissue from the LHMAB area. Now here you can see the left side, and here you can see the right side. The left side is the ipsilateral side. So she took the tissue from the LHMAB area here, and she took some from here, and then do this, and then she processed through, and then she quantified the CART mRNA. So now she's looking at the mRNA, and you find that in the unconditioned control animal, there is hardly any difference between right and left, a CART mRNA on the right side and CART mRNA on the left side. But from the conditioned animals, you find that on the ipsilateral side, there is almost twice the amount of CART mRNA as compared to the contralateral CART mRNA, which means as a result, your electrode which has been stimulating on a particular side, on that side there is more CART mRNA being synthesized, and as a result of that, more CART protein is being synthesized, and we have evidence to show that the entire there is more expression of the mRNA as well as the protein in the cells of the lateral hypothalamus as a result of the electrical stimulation in the adjoining area. These interesting experiments were done by Neha with reference to estimation of CART mRNA. Thanks. Next, please. Neha also did something very interesting. What she did was there is those who are familiar with the brain development. They know that one of the very widely expressed protein in the brain is called as brain-derived neurotrophic factor, BDNF. What is this? This is the protein. What is the peculiarity of the protein? We find that this protein is abundantly expressed whenever a neuron growth is there, whenever a neuron is being stimulated, and if the neuron is undergoing mitosis, as in the hypothalamus. So whenever neuronal activity is going up, it is invariably BDNF is involved. So the question was, if our CART neuronal activity or CART neurons activity is going up, then the BDNF activity should also go up. To test that hypothesis, what Neha did was she took the control animals. She took the LHMAB area of the controlled animals. She took the LHMAB area of the stimulated animals, and here she controlled. So these are the control animals, lateral and you see some neurons with the which are containing CART and the BDNF. But you compare with this figure, you will find that the number of cells which are containing CART and which are containing BDNF has gone up. Means there is certain trophic effect that is being experienced by the CART neurons, perhaps as a result of self stimulation, and because of the electrical current, you see more expression of BDNF, which was again an additional proof that the neurons are CART containing neurons are excited as a result of the behavior that the animals display. And actually she has quantified the number of neurons in the control animals. This is more or less the population of cells which are containing CART and BDNF, but in the animals which have been trained and conditioned, the number of neurons is very high and the difference is highly significant. A very interesting observation. It proves that the presence of BDNF in those neurons proves that the cells are really excited as a result of electrical stimulation. Next, please, madam. But one basic question remained. The question was, okay, if you stimulate here, if you stimulate in the, where is your electrode? Your electrode is in the LHMAB area. But how do you know that the CART neurons really go to the ventral tegmental area? We don't know. I mean, it may be. We have to really test whether the connectivity is there or not. Therefore, then what Amita did was she did what you call as a neuronal tracing technique. She used neuronal tracing technique. This is a very useful, a very basic technique for neuroscientists. Whenever you want to find out, well, we all know that brain are circuits. The neurons in area A are talking to neurons in B, and from B to C, and you have very complicated circuits, and action potential goes all around those circuits, and then they are necessary to generate our understanding, behavior, whatever. To establish the connectivity, what she did was she injected a neuronal tracer, a particular dye which is known to travel along the neurons. So she put dye, which is called as dye-eye. It's very commonly used in. So she injected dye-eye in the ventral tegmental area, and from the ventral tegmental area, and then eventually means, so what you do, you inject the dye today, where in the ventral tegmental area, you let the animal survive for 10 days, 15 days. After that, you take the sections through the LHMFB area. So this is the site where she has injected the dye, and in the LHMFB area she has taken, and there she gets the labeled neurons, which means the dye which was injected in the ventral tegmental area, that dye has entered into the axons, it has traveled retrogradely as far as the LHMFB area, and here you can see the red fluorescence which represents the occurrence of the dye. And then she stained the sections with CART antibodies, and here she was able to show that the CART neurons of the lateral MFB area have projections to the VTA, which means this is a confirmation that the LHMFB area has CART neurons, which is now having a projection or connectivity to the ventral tegmental area. So now we have anatomical basis to say that when you are stimulating the electrode, via electrode, the LHMFB area, not only the CART neurons are stimulated, but they may be projecting to the ventral tegmental area, which we know is very important, a center in the pathway for pleasure. So this anatomical this anatomical study helped us to confirm the circuit that CART neurons from the LHMFB area goes to ventral tegmental area. Next, can I go to next slide, ma'am? Okay, so what she did was, next experiment she did was, this is even more interesting. She had this was done by Amita again. She had two surgeries. One was our typical electrode which goes in the lateral forebrain bundle. This is supposed to and we put there, and then we train the animal, and the animal, once trained, it goes on repeatedly pressing the liver. Then what she did was, she placed a cannula stereotaxically in the ventral tegmental area, and she is now monitoring the liver press activity every day for 30 minutes of the trained animal. And when she gives only the vehicle here is the cerebrospinal fluid, you get about, and then you let the animal into the operant chamber, and you find that the animal presses the liver about 40 times per minute. 40 times per minute. That is with the control, or that is with the vehicle. But now with the same way, she injects CART, and she injects CART peptide at different doses, and she finds that when you give stronger dose of CART, the number of press activity goes up. So when you give this dose of the CART, she finds that it is then, so the number of activity as compared to the control, which is 40, has gone to about 65. Which means that direct administration of CART, which is exogenous CART, which is not naturally occurring into the ventral tegmental area, is also capable of stimulating the liver press activity. Now, as a student of pharmacology, we all are interested in, if the CART is acting, CART must act on its receptor, and if it is acting on the receptor, I should be able to block those receptors or antagonize these receptors, and I should be able to confirm that it is really the activity of CART that is having the agonistic effect. Now here comes a major problem, a very major problem in our current level of study. As on today, there's a problem. As on today, we are not sure about the structure of CART receptor, and there are no CART receptor agonists and antagonists available. Of course, agonist is there, you can use CART peptide, but you have no antagonist. Because there are no antagonists, there's a problem. People have tried to solve the problem by injecting CART antibody. So what they do is, you inject CART antibody here, and in the hope that the CART antibody will take away what all CART peptide is available. So what you are really doing is, whatever is naturally available CART, you are trying to reduce it. And when you do that, this experiment is very standard in literature, and you call it as immunoneutralization. So you try to reduce the amount of available CART peptide which is naturally occurring there. And when you give CART antibody, you find that the number of presses has gone down. So when you give CART, the number of presses has gone up, liver presses has gone up, so reward has gone up. When you give CART antibody, the number of presses has gone down, which again confirms or suggests that the CART probably, if it is released in the ventral tegmental area, it excites the ventral tegmental area neurons, and when they are excited, the animal gets certain amount of reward, and therefore the liver press activity goes up. Can we go to the next slide, please, ma'am? Okay, then what she did was this time she had the animals which had the electrode in the LHMFB area, and these were the trained animals. And this time she took the tissue from ventral tegmental area. Now, what is this ventral tegmental area? We have seen that they are rich in dopamine cells. So she stained the sections and she found that in the control area you have dopamine cells. They are stained with dopamine cells can be stained with an enzyme marker called tyrosine hydroxylase. So she used antibodies against tyrosine hydroxylase, and in the naive animal or in unconditioned animal she saw some cells, but when she looked at the same from the conditioned animal, she saw far more. So you can see you can compare the control with the experimental. This is the self-stimulated, this is the conditioned animal, and there was, so in the conditioned animals, not only number of CART cells has gone up, but the number of dopamine cells has also, dopamine expressing cells has also gone up in the ventral tegmental area. Then what she did was, then she double stained it with CART antibody, and it may not be very clear, but you can see the green fibers here, and those green fibers. So are CART cells there in the ventral tegmental area? No, but the ventral tegmental area is receiving CART fibers, which are shown here in green, and the population of that fibers is also gone up. Means we can say that the cartergic pathway which is there in the lateral hypothalamic area is, and which is sending fibers to the ventral tegmental area, that entire circuit is upregulated. You have more CART in the cells, you have more CART in the fibers, which means in the trained animals, now there is increase in the supply of CART in the ventral tegmental area. She has quantified the data, and she has clearly shown that the number of tyrosine hydroxylase or dopamine cells has gone up, and number of CART fibers has also gone up. This work was done by Amita in our laboratories at Nagpur. Next, please, ma'am. Yes. So going back to the same experiment, so we have electrical stimulation of the lateral LFB area. We know that the CART cells go up, or the electrical excitation goes up here, and then here is the information goes to the ventral tegmental area. And now this time what we did was, we picked up samples from the nucleus accumbens, and we subjected them to dopamine analysis using HPLC electrochemical detector method. Okay, so this is experiment. What is it? There are three interventions. Number one, this electrode is for self-stimulation. The animal is fully trained. There is a cannula that goes to the ventral tegmental area, and there is another cannula which is there to pick up the samples from the accumbens area. Now, in these animals, we did several series of experiments, and the data is now shown in the next slide. I hope you will find this interesting. Can you please go to the next slide? Okay, this experiment was done by Sanjay. So what Sanjay did was, in the ventral tegmental area, he has given control artificial CSF. He has given CART. He has given CART. I'll draw your attention to this bar. He has given CART peptide, and you can compare the control with the CART peptide. And what are you doing? What are you doing? You are evaluating. You have drawn the sample from the nucleus accumbens, and here are your traces. What are these traces? These are the traces of the Hplc traces, and these are the, of course, standard dopamine and standard dopac. And here you give the ACSF, and here you get the two peaks. This is with the ACSF, which is the control, and I want you to ignore the rest, but just compare with the treated one. This is the animal injected with CART peptide directly in the ventral tegmental area, and I just want to compare A4 with A5, and you can see there is a sudden increase. You can see that there is more increase in the dopamine here, and this is dopac, which is a metabolite of dopamine. Both these agents they are increased under the influence of the CART peptide. When you do CART antibody into the ventral tegmental area, the dopamine goes down, which means that in the animal which is behaving animal, so this is an experiment in the behaving animal, you are doing the behavior and you are giving selectively CART peptide, you find that the liver press activity goes up. Not only that, you find that the dopamine release in the nucleus accumbens goes up. You give CART antibody, it goes down, which again indicates that it is the CART which is responsible not only for the behavior that you see, but you can correlate that behavior, that you can say that the behavior is dependent on actually the amount of on the increase in the dopamine as a result of the injection of the CART neuropeptide. Next slide, please, ma'am. Okay, now this is something I'm sure as a pharmacology student you would be interested in this slide. Just as you can place the electrode and stimulate, and the animal can be taught. Can the animal be taught to press the lever to self-infuse the drug? And of course, in the literature you will find there are several studies when the animal can be taught to self-infuse cocaine, amphetamine, opium, all the drugs, the animals are known to self-infuse. So what Gajanan did in this particular case, that he has a rat, and in the rat he has introduced a cannula, and that cannula, now it's not for the delivery of electric current, no, it is for the delivery of an agent. So you can give an agent, any chemical agent, he can give CART. Actually, he has given CART. But before giving CART, he has done a very fundamental experiment. Let us see what happens. Let us see if we can make the animal addicted to alcohol. So what is the setup here? The setup here is a rat. In that rat, the cannula has been inserted. Here you can see the image of the rat on the cannula inserted. This is the picture which was taken after the animal was killed. You can see the passage of the cannula, and that cannula was connected to this polythene tube. That tube goes all the way, and that tube is outside connected to a Hamilton syringe. And that Hamilton syringe, that plunger of the Hamilton syringe is connected to the liver. So now the animal in the beginning, the animal doesn't know, but if you slowly train the animal to go and press the liver, if it presses the liver, then it finds that a certain amount of alcohol, nanoliters of alcohol, is directly released into the ventral tegmental area. Now, if the animal likes it, the animal can be trained. If your cannula is in a wrong place, then the animal is indifferent. The animal doesn't care. But if your cannula is in the right place, and if it gets a delivery of a very small quantity of alcohol directly into the ventral tegmental area, then the ventral tegmental area is excited, it carries action potential to the nucleus accumbens, it will release dopamine, the animal like it, the animal wants to enjoy that experience, so that it will again go and press the liver and have a direct shot. So instead of having alcohol in a regular glass, you are delivering alcohol directly into the ventral tegmental area, and you are teaching the animal how to self-administer. Here my students like to call that liver as a bar, pun intended. Okay, so and all these experiments were done by Gajanan, and can we go to the next slide, please? So this experiment was done by. Okay, now what Amita did was, she took this is interesting. She took those animals which were trained to self-administer. These were, look at the previous video. These are the animals which have been trained to self-administer ethanol directly into ventral tegmental area. Now, what Amita did was, she took those animals, she gave them a wash period of about 7 days, and she said, let me see in these animals, if instead of alcohol I give CART, does it work? That was a very simple question she was asking. So what she did was, so she is comparing actually the two livers. One is the active liver and the inactive liver, and she found that when so what is an active liver? When the animal presses the active liver, alcohol is delivered. Inactive liver, nothing happens. And she compared how many times the animal presses the inactive liver, and you can clearly see this is inactive, this is like a control, and the animal presses the active liver far more. It's about 15 times over a period of a few minutes as compared to the control. And here is the quantification of the data, and here you find that cart. So this is a 50 nanograms of cart, you get this response. 100, you get so much response. And she could actually optimize the dose of cart when that cart was directly injected. The animal was trained to self-administer cart directly into the ventral tegmental area, which means cart was capable of generating reward into the animal, and that the cart was acting specifically in the ventral tegmental area, and that experience resulted into the animal getting a sort of conditioned to that procedure. Next, please. Next slide, please. Yes. Now, what Amit was doing was something very. He said, "You see, Amit was doing something very interesting. I actually have a lot of appreciation for that." Okay, what Amit said that, well, cart is changing. We know that cart is changing in the lateral hypothalamus. Cart is changing in the ventral tegmental area. He said, "Let me see where else cart is changing." He just took controlled animal. He took control animals, studied the distribution of cart, and then he took the trained animals which were trained for self-stimulation, and he compared them, and he found that not everywhere, but there were certain specific areas where there was a profound change, and that one particular area which he could identify was paraventricular thalamic nucleus. Now, what am I talking is a group of neurons. Okay, good. Where is that neurons? It is in the thalamus. What about that neurons? The neurons do not contain cart, but they have a rich innervation of the cart coming from cart cells. Now, then we went into literature and we found that the cart neurons of the LH, which we have already shown, they also innervate this area, which we call as paraventricular thalamic nucleus. And then he showed here, just see here in the control, are cart fibers there? Yes, they are there. But just compare with this, and here he found that there are far more cart fibers which are located here. And then he saw in the paraventricular nucleus, he wanted to find out whether paraventricular neurons are also excited as a result of these, as a result of conditioning. So you use CFOS, and he found that the neurons of the paraventricular nucleus, they do not contain cart, but they receive cart. They also express more CFOS. There is double labeling which are showing. So the neurons of the cart, the neurons of PBT which do not contain cart are all red dots, and the green that you see are the cart fibers which are coming from the LHMAB. So he wanted to show that the nucleus of the paraventricular, the neurons of the paraventricular nucleus, they are also getting excited as a result of the self-conditioning protocol, and that the cart innervation is going up, and the cells are showing an increase in the expression of CFOS. Yes, next please. So then we wanted to test another possibility. Do the neurons of LHMAB, they project to the PVT? We went to the literature and we saw, yes, there is already studies which show that there are cart neurons. Of course, they are there. Do they project to PVT? Yes, they do. But do they have a role in reward? We don't know. If they should have a reward, then they must have a connectivity to the nucleus accumbens. What kind of connectivity? We don't have much information in the literature. Therefore, then we decided to explore this another aspect of the cart circuitry. We have already shown that the cart neurons have ventral tegmental area and dopamine go to the accumbens. Now here suddenly we had evidence to suggest that there is an alternative pathway, and we decided to go deeper. So we wanted to explore the hypothesis, which is glutamate as a neurotransmitter. So one question was, do the cart neurons which are located here, they go to the PBT according to the literature? Do they talk to the glutamatergic neurons? And second possibility, do these glutamatergic neurons communicate? Do they have any role? Questions we wanted to pursue, and therefore I'll take you to the next slide. Oh, we have one basic thing is, whenever you say, you have to show the connectivity. Connectivity of what? Connectivity whether the PBT neurons they really project to the nucleus accumbens. To that end, what Amit did was Amit injected DI, which is a neuronal tracer. You inject today, you wait for 15 days. After 15 days, you take the sections in the paraventricular thalamic nucleus, and we find that the neurons are there, which means what? There is definite connectivity is there. Okay, are those cells are the cells which are labeled or which are communicating with accumbens? Do they have card there? Yeah, they have card there. So here is a label. So we are looking at the neurons of the PBT. Number one, those PBT neurons have exons to the accumbens, and they are card. And where is this card coming from? The card is coming from LHMAB area. So actually this is we are showing PBT. What about PBT neurons? On what one thing that they are sending exons to the accumbens. On one, they are receiving exons from the cartridge exons from the LHMAB area. So it is a sort of hub which is on one hand getting input from the card neurons, and on another it is giving output to the nucleus accumbens. Next, please. Okay. Now, but you have to prove, you have to prove that the neurons really contain glutamate. How do you know that they are glutamatergic neurons? For that, there is a marker which is actually called as okay. Let me explain this very briefly. Glutamate neurons have vesicles. In the vesicles, there is neurotransmitter. That neurotransmitter is glutamate. The covering or the membrane that is going around the vesicles has a special kind of protein which we call as a glutamate transporter. Glutamate transport is different type, glutamate 1, 2, 3. People have shown the occurrence of glutamate 2 in the neurons of PBT, and we also tried to show. So what Amit did was he stained the sections through PBT using the antibodies against glutamate V-glut, that is vesicular glutamate, and he tried to co-label it with CART. So what are we doing? We are looking at the PBT neurons of the control animals and of the conditioned animals, and you can clearly see the difference, less CART here, more CART here. Then he stained the same sections with the antibodies against V-glut. So some glutamate is there, far more glutamate is there, which means the neurons of the paraventricular thalamic neurons are also excited, and they are expressing more glutamate as a result of the conditioning experiment. And here you see that here you have the co-labeling. Here is the unconditioned animal, here is a conditioned animal, and you can see more innervation of the CART. So CART fibers are coming from the LH hypothalamus, they are terminating in the PBT and on the PBT neurons which have glutamate as a neurotransmitter, and you can quantify the data, and the quantified data has been shown here. With what are we studying? The PBT neurons, their response to conditioning. What has gone up? Glutamate has gone up. Glutamate has gone in the cells, and the innervation from CART neurons has gone up. Can I go to the next slide, please? Okay, this is then what it did was, look, if the glutamatergic neurons are communicating with the nucleus accumbens, if glutamatergic neurons in the PBT are communicating with the nucleus accumbens, and if they are using glutamate as a neurotransmitter in the nucleus accumbens, you must have receptor for glutamate. And we know we know that AMPA is there, kinase is there, and NMDA is there. So we could get antibodies for glutamate receptor. And then what Amit did was he stained the accumbens region. In the control he had some signal, but he had much stronger signal in the accumbens, in the nucleus accumbens with reference to NMDA. So what Amit is trying to show, that in the nucleus accumbens, even the glutamate receptor NMDA receptor 1 population has also gone up in the neurons of the nucleus accumbens as a result of the conditioning. So as a result of conditioning, we can see a series of changes in the LHMB area, in the ventral tegmental area, in the nucleus accumbens, in the paraventricular thalamic. So all the different points in the nodes of the circuit, we are showing using proper markers, we are able to demonstrate the significant changes at each node. Can I have the next one, please? So we are just again going to the original experiment where we are just counting the number of liver presses. But here what we are doing in the ventral PBT area, we are directly giving card. So in the PBT area, you give the vehicle, nothing happens. No, not nothing happens. You get a liver press about 36 times. But if you in the PBT area, if you directly give card, then you find that the number of liver pressing has gone up, which means CART is active in the paraventricular thalamic nucleus also. So we are showing two sides. One is CART is acting in the ventral tegmental area, stimulating reward. CART is acting in the paraventricular thalamic nucleus, and again promoting reward or in the rewarding behavior as quantified by the number of liver places. Next, please. Okay, so we again, if that is happening, if the reward is happening, we must monitor the amount of dopamine. So this is an experiment done by Sanjay. What he did was he has this electrode in the LHMF area. The animal has been conditioned, and in the PBT region, he gives CART, and then he collects the samples from nucleus accumbens and he subjects those samples for the HPLC so that he can evaluate dopamine. And in the next slide, he presents his data. Next, please. I'll just draw your attention to these two curves. This is the dopamine trace. Here you can see in the control animal, I just want you to compare this peak. I want you to compare with this peak. This is when you give the vehicle, this is the amount of dopamine you get. When you get CART, you can see the increase in the amount of CART. I made a mistake. Increase in the amount of dopamine when CART was given in the paraventricular thalamic nucleus. So in the paraventricular thalamic nucleus, you give CSF, you get so much release of dopamine in the nucleus accumbens. You give CART in the paraventricular thalamic nucleus, you get an increase in the dopamine, and this has been quantified. Area under the curve has been quantified, and Sanjay has clearly been able to show that administration of CART in the paraventricular thalamic nucleus gives rise to increase in the amount of dopamine, and we know that increase among the amount of dopamine in the nucleus accumbens, of course, is very necessary for the generation of reward, which again is responsible for increasing the number of liver presses per unit time. Next, please. Okay, so here this is my summary slide. I'm going to conclude my talk in next 2 minutes. So what do we conclude, or the take-home message is there in the LHMFB area, FD hypothalamus, we have a population of CART neurons. Some of the CART neurons they project to the ventral tegmental area. And under natural conditions, we believe that it is excited. So maybe when you have food or when you have water, these neurons are excited. They use CART to excite the paraventricular, the VTA neurons, which contain dopamine. Then they carry dopamine. When they are excited, they secrete dopamine. When they dopamine, there is reward. Alternatively, the CART neurons here could also talk to the ventral thalamus, to the paraventricular neurons of the thalamus. When they are excited, they could talk to the glutamatergic neurons here in the paraventricular thalamic nucleus, and these glutamate neurons could then act on the neurons of the nucleus accumbens, which have receptors for glutamate, NMDA receptor type 1, and when they are excited, they will again generate reward. And our study broadly shows the importance of the neuropeptide Y, which is located in the neurons of the lateral hypothalamus, and their role in modulating the activity of the reward pathway, which is traditionally very well known to start from the ventral tegmental area and end in the nucleus accumbens, and promote the release of dopamine. I think I have come to my time is good. I hope I'm good, and I must thank, sure, I should not forget this slide. This is a contribution from 2 labs. Most of the work was operant conditioning and training. All that work was done in Nagpur. Our lab in Department of Pharmaceutical Sciences. The PI there is Dr. Kokre. He under his lab, these are all his PhD students. Here is Amit. Here is Amita. Here is I'm sorry, here's Gajan. Sorry, I made a mistake. Gajanan, here is Amita is here, Amit is here, and Sanjay is here, and here is Amol Sakharkar. He is in Pune University, and associated with him is Sneha. She helped us with mRNA analysis, and I I thank most all this work was done by these members of my earlier lab, and it's only because of them. I give all the credit to my students. They have done this. Please let me tell you, the training the animals is extremely cumbersome. Surgery often fails. It's not always very successful, but painstakingly and with lot of patience they have done it. And thanks to them for coming up with this interesting data. Now I conclude my talk and thank you very much for your patience.
Thank you so much, sir, for delivering such a illuminating session on CART neuron and their screening. I would like to request Dr. Mrinal Sanai to briefly summarize the session. Please, madam.
Thank you, Ruchira madam, and thank you, Dr. Subhedar sir. It was such a wonderful session and such a critical topic you have explained in a very simpler manner. And you started from the role of CART in the satiety centers. Then you have even talked about the ventral tegmental area and medulla oblongata, nucleus accumbens, and how the stimulation of the ventral tegmental area can lead to the increase in dopamine levels and therefore the reward. You have explained the principle of the reward model also clearly. You made the basic concepts of the neuropharmacological study to assess the reward model very clear, and then it was very easy for us to understand the further neuronal studies where you have presented the results for upregulation of the CART and the role of the C-force to check the expression of the lateral hypothalamus neurons. Recording of the CART mRNA expression also you have shown. You have highlighted the importance of the neuronal tracing techniques to establish the anatomical data, to establish the connections of the CART neurons to the ventral tegmental area. You have also given us a exposure to the challenge that the CART receptor structure needs to be elucidated. It is still a challenging area. A very interesting was a technique which you have shared with us on conditioning of animals for the self-administration of the drug, and that was really interesting to know, and you have shared that data. Not only that, but how one lead leads to the other. That means you have established the role of this ventral tegmental, lateral hypothalamus medial brain role and its connection to the nucleus accumbens. But again, your students and you have moved further and have tried to establish the to probe the connections of the paraventricular thalamic neurons to the nucleus accumbens, and that has given you the newer area to develop of the glutaminergic neurons and what is their role. And then there is a hub you said that is from the lateral hypothalamus medial forebrain to the paraventricular thalamic neuron, and then paraventricular thalamic neuron giving the input to the nucleus accumbens. So it was so beautifully you have presented and made us to understand such a critical topic, and I'm sure, sir, that it was a reward for all of us. So right now, if you check the expression of the CART, you will get a clinical data also, with the attendees' brains. So it was really rewarding and very, very beautiful session. Such a critical topic you have explained in a very simplified manner. Thank you very much, sir, for that. And now we open the session for question answer. I will just go through the chat box and I will see are there any questions. I will request the audience to please post your questions. If you want to ask the questions, please post it in the chat box. And those who want to communicate with sir, I will offer a mic. Please raise your hand. This is a lifetime opportunity for all of you to communicate with sir. There is a question by Vaibhavi Peshattivar. Sir, that since behavioral experiments are tricky, since there can be huge variability in the behavior of animals, how to handle such situations, or what could be an ideal number of animals to be included in the experiment to make up for this variability?
This is a very good question. And I'm sure this is a very good question. I'm sure all behavioral biologists and pharmacologists have faced the problem. because the behavioral data is very difficult to get clean data. So it's a very good question. Very difficult. I'll use the word very difficult 10 times. It's very difficult to get clean data. Okay. But in the case of in our experiments, in our experiment, there is a litmus test. What do I mean by litmus test? You do the surgery, you place your electrode in the lateral hypothalamus, you give 7 days to the animal to recover, and on the 8th day, you start training the animal. Now, our laboratory norms are, if on the 12th day, if the animal starts learning, means even if the frequency of pressing the active lever is say if the inactive lever is 5 times, and if the inactive lever is 20 times and more, then we come to the conclusion that my electrode is in proper place and the behavior it shows is really meaningful. If it is less, then we don't use the animal. Because of the behavior, because of the liver press activity is such a clear parameter that if the liver press activity is not there, which means it has not been trained and it can never be trained because of my mistake that the liver is in wrong place. So there are a lot of rejections. Not every animal can be trained. The problem is sometimes only the animal is partly trained. Then what do you do with it? Okay, so we have to do far more number of animals. Okay, and decide a cutoff line that if the animal's performance is not up to this line, then we will reject the animal, and if it is higher, then only we'll accept the animal, and then only we use that animal for further injections like various injections through the cannula, etc. But because of clear-cut behavior, we have set a threshold that any animal which shows liver press activity only above this and can be trained in 3-4 days, then only we'll take it. Otherwise, we'll not take that animal and we'll not use that data. I don't know. I have made an attempt to answer that question. Are you okay? Is she okay with that answer?
Yeah, sir. I think.
No, you get confirmation from her.
Yeah. I'll just waiting for a confirmation. I think she's
okay.
Sir, I have one query that since you have probed that there is a lateral hypothalamus medial forebrain neurons connected to the nucleus accumbens, and there is also the role of BDNF, which you have said that you have derived. So can this model be also useful for the screening of antidepressant drugs, because BDNF is becoming as one of the targets for development of the antidepressant drugs also?
Yes, madam. For the actually that lab is also being pursued in our that line is also being pursued in our lab. What student has done is he has used what they call as deep brain stimulation. Okay, and which is slightly different from this. But in deep brain stimulation, we have shown that deep brain stimulation of certain, it's also there in the literature. Deep brain stimulation of certain areas gives relief from depression. Okay, that is very well known. But and I'm also sure that in those conditions the BDNF will be expressed. It will be expressed. I'm absolutely sure it will be expressed, but we have not done it in our study. And if the literature may be there, but I'm not aware of that literature. But I'm sure literature will be there, which tell you whether following deep brain stimulation of certain areas, what happens to the expression of BDNF. I do not know, but I am absolutely sure literature will be, the information will be there in the paper somewhere.
Thank you, sir. Thank you very much. There is a question by our student, Rushikesh Kulkarni. He says he is thanking you. He's saying that it is a very interesting session, but he has one question, that how will the mechanism of action of CARD antagonists after this discovery differ from those drugs which are used to treat addiction?
Okay, this is now look, the point is very valid. The point is very valid. Can CARD be used to treat? The answer to that question is there is a big gap. We don't have sufficient knowledge. Number one. Okay, let me answer your question. Number one, you will have to first find out the receptor for CARD. If you we are stuck, you know, entire community, scientific community, there's a big community there in Australia, USA, everywhere. All the people are, they have only, whenever they meet, they only talk of one thing: could you find the receptor? There's no receptor. It's now almost 25 years, there is no receptor. Number one, so you will have to find a receptor, and second, you will have to find out agents, non-peptide agents, which will act on the receptor. Because you see, you can't give a protein molecule like antibody into the brain. You will have to give some molecule, some agonist which is non-peptide, but which can be taken by a tablet or injected, which will go as far as the brain, cross all the blood-brain barriers and act on the receptor. But such peptidomimetic agonists are available for, say, neuropeptide Y, or for MSH, they are available. But for CART, although it's a very interesting strategy to build, there are two major blocks. Number one, you have to get the CART receptor, and then you have to get non-protein agonist for those receptors, which can be taken as a tablet. If you can come up with, if you can cross these two hurdles, which I understand are very difficult, if you can cross these two hurdles, then we are ready to answer your question. It is too early to answer your question, but yeah, I think I have conveyed my point. Thank you.
Sir, just a thought came in my mind when you were discussing that, okay, let's accept that there is no characterization of the CART receptor till date. But is there any possibility that these CARTnergic neurons are acting on the existing endogenous receptors, maybe of a dopaminergic or a glutaminergic, on some allosteric site and triggering?
Madam, actually, the existing studies show that there are the receptors are there. Okay, I mean, not that we have not made, the receptors are there, the CART acts on that. We also know that they are via cyclic AMP mechanism. Okay, they are G protein coupled receptors, so we know a lot about it. Okay, so there are evidences which shows that the CART receptor is there. It is on this particular cell. The cascade of events or events intracellularly, these steps may be happening. That's all there. But you, that's not enough. I mean, you need to extract that protein and you need to determine that structure. So not that we have not made any progress. Some progress is there. Okay, so it's just like we have a receptor for NPY. So there is receptor is there. It is G protein coupled, but that's not enough. You have to get that protein. You have to purify that protein. And all that work is not successful still.
Okay.
So yeah, so just as there's a protein for so many peptidergic neuromodulars are there, so that we believe that it's there. Okay, but somehow we are not getting that molecule in our grip.
Okay. Thank you, sir. There is a question posed by Dr. Vrish Ashwalayan, that is,
Yes.
how individuals' variation in behavioral animal studies can be overcome. How individual, same that what is individual variations in the behavior of animal during studies can be overcome.
Again, the same. This is very much like the first question, you know. And I'm sure I'm sure anybody who has worked on the behavior comes across this problem. You have to have enough number of animals and the statistical power. You see, even when we submit our papers to the journals, this is a question which is very often asked to us by the referee. He says, how do you make sure that your behavior, that the behavior of the animal you are saying is not? Yeah, but we can convince by, if we have sufficient number of power, a sufficient number of animals and the statistical power. Okay, and then by using methods, we can say that, well, my behavior can be relied upon, and then it can be reproduced, etc. But mainly, the answer to your question is enough number of animals and statistical power. If you can use these two tools, you can convince your referee.
In line with that, there is another question asked by our faculty, Ms. Rupam Raut, who is Assistant Professor in Department of Pharmaceutics and pursuing her PhD, that she is asking that n equal to 6, is it okay, sir? Means the sample size equal to 6 animals in one group. Is it okay to conduct these types of studies?
Well, okay. Now you can again debate forever. Okay, whether and on this point, I have been harassed by a lot of referees. Okay, it's not that they have me. But if you see, let us go by studies. You see, most of the people use 6 animals. Some people use 8 animals. I have also used 8 animals. But if there is consistency across the group, if there is consistency across the 6 individual animals in your group, okay, then by using right statistical power, I would say that I mean, let me see, if you have a group of 6 animals and there is only one outlier, you can still use it, okay, but if there are 2 outliers, you cannot use it, okay, and if there is not a single outlier, then you are good about it. Okay, so the answer to your question, if there are no outliers and if there is consistency within the group, 6 is a fairly acceptable number of animals.
There is a question by Vaibhavi Peshattiwar that can dye used in this study for determining connectivity be applied for studying neurogenesis and their migration? Can dye be used for this?
No. For neuro regeneration, if she is talking of neuro regeneration or degeneration, what is it? Regeneration or degeneration?
Neurogeneration.
Neurogeneration. Okay. Now let's see. In the case of mammals, the neurogeneration happens in 2 places. It happens in the hippocampus number 1, and it happens in the subventricular zone. These are the 2 places. If you want to see that happen, then we have better techniques. Nah? You can use what is the technique? I'm forgetting the name. Can somebody refresh me? You see, you inject that molecule. It is not BDNF. I'm forgetting. There are special markers which will show you which will specifically show you the cells which have been reproduced right now. Okay, there are very special markers, and so what you do is you inject that marker. My memory is going down. I'm forgetting the name of that agent. I have used that agent 20 years back. Okay, so that agent, you can inject that agent, and that inject is incorporated into the DNA, and so the newly born cells can be specifically labeled with that. If you want more about it, you send me a mail. I'll answer your question, and I'll also refresh my memory and answer your question.
He's asking, is it BRDU?
It is. I'm sorry. I'm referring to BRDU.
Yeah, it is.
I forgot about. Sorry about that. I forgot. I'm talking to BRDU. I mean, use BRDU. Why do you want to use BDNF?
Actually, Vaibhavi is a student of Institute of Chemical Technology.
Okay.
She's under guidance of Dr. Sadhna Sathe.
Okay.
Work is also on the neuropharmacology.
Yeah. Sure. Oh, you can know in regenerating neurons. Of course, you can use BDNF because BDNF is also a marker for regeneration. She's right. You can use BDNF for regenerating cells. But if you just want to comment on dividing cells, then use BRDU. No?
Oh, I will just check, sir, if anybody has raised hand.
Sure.
No, sir. There are no hand raised.
Okay. Good.
I think I have covered all the queries in the chat box.
Yes.
So, sir, should we conclude with the session? It was really an informative session, and it was. I'm sure that this session will be motivating and will trigger the research into the research for the faculties attending your session in their areas of neuropharmacology. So thank you very much, sir. For
ma'am, I want to make one point. Please let it be known to all the participants that if any one of them is interested in stereotaxy and the different brain interventions, and if they at any stage they want to contact me, you can give them my email and I'll be happy to talk to them, because I'm interested in drawing more and more pharmacology people into neuropharmacology. It's an exciting area and we need more people to work in this area. So I would be, and at my age, I think that is more important. Okay, so thank you very much.
KMK will be honored to collaborate with you, because there are many such projects going on in our laboratory also.
Sure.
I will surely give your contact to the other scientists who are who want to.
Feel free, madam. Yes, sure, madam. Sure.
Thank you very much. Thank you very much, sir, for your valuable time and delivering such a wonderful information. Thank you.
Thanks, ma'am. Thanks.
Over to you, Ruchira.
Yeah. Okay. After having such a magnificent session with Subhedar sir, now it's time for tea break. I request all the delegates to resume back at 11:15 for the second session of the day. Thank you, everyone.